Our session before we take our next scheduled break is Dr. George Kuchel to speak on immune function. Dr. Kuchel's research has focused on precision gerontology and geroscience-guided interventions designed to enhance function and independence in older adults by targeting heterogeneity in aging-related declines involving host defense, mobility, cognition, and bladder control. George, the stage is yours.
Thank you.
Oops, sorry, you have a...
Is my microphone on?
Yeah, and then down is forward.
Down is forward. Remember that down is forward. I'm sure I'll get confused. It's my pleasure to be here among many of my favorite people in one of my favorite cities. And my talk is going to be a little bit different from the other two earlier ones for a number of reasons. One is, as Jamie alluded to, the topic of immune aging in the context of gerotherapeutics could be a session in itself. Secondly, what I really want to, because of that, I really want to highlight what's different about measuring immune aging in the context of clinical trials, because the way that most clinical trialists think about these issues is you do a superbly well-designed clinical trial, you collect lots of serum, and then you do a whole bunch of freezer studies afterwards. And that clearly is not going to work for this kind of a context in terms of gerotherapeutics. We need to give these thoughts, we need to think this out carefully beforehand, also in anticipation of new technologies that will become available as the studies are underway. And finally, I also want to highlight the fact that these trials will absolutely, as we heard earlier today from Nick Shork, these trials will have to be done in people who are not perfectly healthy, who are in that sweet spot between being healthy and being disabled and significantly disabled, so they have the accumulation, being at risk of developing new chronic diseases that ultimately these gerotherapeutics will prevent. So I have some disclosures, but none are relevant, no relevant commercial disclosures. So moving gerotherapeutics from an idea to a reality, we heard a lot about the fact that aging is an incredibly heterogeneous process, illustrated here in the context of physical function, as Dr. Cummings just showed earlier. We now know, thanks to a lot of the hard work done here, we now know a lot about the biology of aging, all these different hallmarks, not just how they each function and what their nature, but also how they're all interconnected. And that's wonderful. We made a tremendous progress over there. And that has led to the idea that we can then, this is data from the Olmsted County accumulation of chronic diseases, 3 or more chronic diseases. The frequency goes up significantly, exponentially with aging. This whole idea that using gerotherapeutics, we're going to be able to push that back. And I think it's a wonderful concept. We're incredibly excited about it. Has lots of potential. But in terms of how that's actually going to happen, in terms of how our knowledge, in terms of the biology of aging, and our knowledge of what happens to older adults as they age, how that's going to be translated and connected, and how these gerotherapeutics are ultimately going to work in humans is still work in progress. So the traditional view of disease biomarkers comes from this Institute of Medicine report from 2010, which basically says a biomarker is a characteristic such as a cholesterol level that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to intervention. So some of that is still true today, and it certainly applies to diseases such as you see here, for which you have a single risk factor or predominant risk factor, such as many inherited diseases. You can have progression to early disease or advanced disease, and I think there it works pretty well. The problem is that most disease processes studied in isolation, but particularly when studied in aggregate in a single living person with aging, do not develop in this manner and demonstrate tremendous multifactorial complexity at all levels that I'm going to just talk about very, very, very briefly here. This goes back to a paper that a number of my colleagues and I published a few years ago, just to highlight that in the context of geriatric syndromes, which ultimately everything we talk about today could be classified as a geriatric syndrome, which is a multifactorial condition of aging, for which aging is a major risk factor and therefore occurs increasingly with aging, typically cutting across traditional organ systems. That's the other thing about aging, is that diseases of aging do not respect boundaries between different tissues, between different organs or professional disciplines. And one thing that we see is that we see that everything that occurs with aging is highly, highly multifactorial, as illustrated here, with a real important role for shared risk factors. And the biology of aging, the big advance that occurred since we published this paper, that was at a time when the word geroscience didn't exist, is that biology of aging, in fact, represents one of these incredibly important shared risk factors between all these different chronic diseases or geriatric syndromes of aging. So one of the challenges has been, and this is from a graphic that a number of my colleagues, including Jamie and others, have published in a textbook of geriatrics and gerontology, is that there's deep skepticism, and I want to emphasize deep skepticism amongst clinicians who take care of older adults that therapeutics are going to work. And the reason they're skeptical, well, it's partly because we've now cured Alzheimer's disease about 10,000 times in mice. The other skepticism is the fact that when everything is so highly multifactorial, how could possibly a single pill? Now, people accept exercise. Yeah, that could have an effect because the exercise is incredibly pleomorphic effect in almost everything you look at across the human body. But how could a single drug have such an effect? So this is just to highlight some of these issues where what you have in a typical individual on the far right, you have multiple geriatric syndromes and various chronic diseases of aging. And then you've got these hallmarks of biological aging. Some are a little bit upstream and some much further upstream. But we now know they're all interconnected. Now, you may be interested in one particular hallmark of aging, but it interacts with all the other hallmarks that your colleagues are studying. And so the thought is that if we're able to target this one hallmark of aging, particularly if it's upstream, through its interactions, the effects, downstream effects on other hallmarks, and then the shared risk factors for all these chronic conditions down all the way down to the far right, it's going to have this broad effect. It's a wonderful concept. It may be true, hopefully it'll be true, but really this is really the holy grail of what's going to determine whether all of this ultimately works or not in older individuals. So biomarkers for geroscience-guided clinical trials, I have to go back. I promised myself, I haven't talked about the TAME trial for a couple of years now. I'll say I'd like to nominate TAME trial as the best designed study that never took place, but hopefully will. Pardon me? Not yet. Not yet. But I want to go back to some work we did back then that involved Jamie and Nir and many others in terms of designing the TAME trial, but also coming up with, and that's my illustration of TAME trial, the little engine that could, and hopefully ultimately will. With criteria that we came up in this paper that was published in Geroscience in 2018, which is a framework for selection of blood-based biomarkers for geroscience-guided clinical trials. And I should say the paper is a little bit mislabeled because I got so many emails afterwards, people asking me why we didn't pick their favorite biomarker. It should have been labeled for geroscience-guided clinical trials called TAME, because every gerotherapeutic trial is going to be different, depending on what population you study, depending what your intervention is, what your goals are, etc. So going back to that, this is just a summary of what we came up, and this is still work in progress in many ways for reasons that have become obvious, in terms of role of immune aging within the XPRIZE competition. So the goal here, as you've heard, immune function is going to represent one element of the important triad that you're hearing about today. Improvement from baseline that exceeds personalized response thresholds in 2 out of the 3 biospecimens-based biomarker categories as measured by central XPRIZE laboratories. So first one, specific assay decisions will be determined in 2026. So still a work in progress, but may include cytokine multikine assays. I'll talk about that. Some immune cell compositions such as IMAGE that I'll talk about as well. And I think what's particularly important is some ex vivo naive immune responses to a new stimulus, which I think is going to be particularly important to uncover resilience, in this case, immune resilience. So when people think of biomarkers, you always begin with what's easy and what everybody else does. So people talk about circulating humoral biomarkers for geroscience guided clinical trials. And this is what we came up with for TAME trial. Again, I'm not going to go through this in detail. The paper is easily accessible. But the point is we selected by number of criteria, and they're listed over here. Number one, measurement, reliability, and feasibility. We didn't want to be inventing new science. We didn't have 50 years to validate biomarkers. We wanted to begin with biomarkers that are already validated. Relevance to aging. If you're testing a gerotherapeutic, you have to be measuring aging, biological aging, I should say, in some way. And a robust and consistent ability to predict all-cause mortality, clinical and functional outcomes. So immediately you have a problem if you think about it. You need something on the one hand is doable, number one. Number two, it's relevant on aging, so it's relevant to biology, which is typically done in a lab, and then has been validated in terms of outcomes in large, well-conducted observational or clinical trials. So there then you talk about epidemiology. So immediately you're trying to link these fields that typically have not worked together. And finally, we wanted them to be some evidence that the biomarker actually responds to the therapy being tested, which is why every intervention trial, every gerotherapeutic trial, ultimately may have some shared biomarkers, but will have to have some unique biomarkers that are unique for that particular study. So that's what we selected. I'm not going to go through it in detail, but the fact is, when you look at that in terms of immune aging, and everybody's familiar with this latest illustration of the hallmarks of aging, the fact is that the ability to obtain measurements for all of these, so any one of these can be measured in a wet bench lab, particularly if you work with more simple animal organisms. But the ability to obtain measurements using serum or plasma, and they're far from equivalent that we can talk about later, that can provide deep insights into underlying biology of aging is actually quite limited. And it's pretty good in terms of chronic inflammation. I saw Deep Dixit here, and it measures whatever we think inflammation is. So it's a bit of a circular argument, but we can certainly measure what we think inflammation is in peripheral blood, and that is highly predictive of clinical outcomes. It's also pretty good at measuring deregulated nutrient sensing in terms of measuring glucose levels and other measures of metabolism. It's okay in terms of measuring cellular senescence, assuming you do it right, and there are many, many caveats that I'm sure others could answer here. It can measure mitochondrial dysfunction indirectly. We talk about serum or plasma. We're talking about cell-based assays. Genomic instability. Again, if you're able to measure free circulating DNA or RNA. Similarly for telomere attrition and many of the others. The point is that for the vast majority of these, it's pretty limited in terms of getting deep insights into the biology. But that's what you typically have in the context of human subject studies. There are some new developments. We heard a little bit from Jim Kirkland about work being done looking at urinary biomarkers, which is very exciting. There are also some new techniques for measuring interstitial fluid using relatively minimally invasive patches, which is very exciting as well. But at the end of the day, blood or blood components is really the most likely source for most biomarker measurements. So the issue of resilience, and why is that important? This is a graphic that accompanied a white paper that Ann Newman and Evan Hadley from NIA and I worked on a number of years ago that came together on physical resilience. What it highlights is the fact that biological aging is a driver not only of chronic diseases, but also of diminished resilience, impaired function, overt disease, and multimorbidity. And of course, multimorbidity itself is a driver from compromised resilience. So there's lots of evidence, and you can see it clinically. The clinicians here will know that your patient may be doing perfectly all right if you don't challenge them, if they're simply sitting in a chair and not doing anything. But the moment you stand them up and you measure their blood pressure standing, or you put them on a treadmill, you will detect abnormalities. So resilience is absolutely important. And this is a seminal paper now published by Joan Manick. And I can't believe, it's 10 years now since this paper came out, 2014. And this was a seminal paper which showed that, administration of rapalogs, mTOR inhibition community-dwelling older adults living in Australia and New Zealand improved responses to influenza vaccine in terms of antibody titers. And most importantly, she showed really nicely that PD-1 programmed death cell marker decreased significantly in CD4 positive T cells with mTOR inhibition as shown in the treatment group at the bottom. So it's possible using circulating immune cells to get at the biology of aging, but it's not easy. It's not easy, and this is obviously a lot more progress needs to be done here. I also want to give a shout out to one of my colleagues, Jenna Bartley, who's been funded by FR and the TGN to look at effects of metformin on many different aspects of cell biology and biological aging that can be studied in peripheral blood cells, including immunometabolism, including glucose processing, and including show some. This is a population of non-diabetics, I should say, in terms of finding some changes, decreases in some terminal differentiation markers in a subset of T cells. So I also want to highlight that heterogeneity of immune resilience is an important concept. So it's important to think about if you're going to challenge the immune system, and we're proposing that it could be done through vaccination. It could also be done through ex vivo studies of peripheral blood cells that are examined. In the document, we talked about exposing to an antigen that the individuals have never seen before, such as some of the yellow fever, for example, which at least in this part of the world, most people have never seen. But I also want to highlight the fact that these responses are incredibly heterogeneous at many different levels studied. I want to just illustrate one example is a paper we published recently with colleagues at Jackson Lab, Jacques Bachot and Kuchel, showing that basically if you take 2 pneumococcal vaccine, and I think many of you in this room have received either the Prevnar or the Pneumovax vaccine, which are commonly administered. They're very different vaccines. Prevnar is a conjugated vaccine. Pneumovax is a polysaccharide unconjugated. The way that people respond to it is very different. Also very important, women and men respond very differently to Prevnar, okay? And the baseline predicts are very different. I'm just mentioning that it's something to consider as you design this. Important limitations of challenges and opportunities. Obviously, getting blood is not non-invasive, but it's minimally invasive. We do it all the time. Has many, many opportunities. I want to urge everybody to think about, in designing your trial, to absolutely collect peripheral blood cells. It's often forgotten because it's a lot of work, and this, peripheral blood cells are often discarded. People just collect serum and plasma. I don't think that's going to be sufficient here. One important limitation to consider here, this is out of a paper that came out in PNAS in 2023. The numbers are not important. It shows the heterogeneity of peripheral blood in humans. What's really important is the immune cells that we measure in peripheral blood represents about 1% or less of all the immune cells in the body, okay? So you're sampling less than 1% of all the immune cells in the body, and we don't really know what that tells us about immune or biological aging at the level of individual tissues. That's really something that is an area of high priority in research. And I also want to give a shout out to the CENNET network. And for those of you who are not familiar with it, this was a major investment made by NIH Common Fund, the Cellular Senescence Network. Many of us here are involved with it. And just a picture of the centers that are involved in it, including, I should add, a number of European centers, basically to create high-quality maps of senescent cells within human tissues, beginning with healthy human tissues across the lifespan, gradually moving, hopefully, with the next funding cycle to more diseased tissues, the kind of work that needs to be done before we really be certain that what we're measuring in peripheral blood actually reflects what occurs at the level of individual tissue. So I want to conclude now in terms of, first of all, I think measures of immune aging may and will hopefully, I think, will be an essential component to guiding gerotherapeutic trials. Humoral, serum, or plasma-derived biomarkers are easiest and best validated, yet they offer more limited biological information. and I'm happy to answer questions later, but serum or plasma are absolutely not the same, particularly with aging. They tend to differ quite a bit. Cell-based immune measures and offered potential for deeper biological insights and need to be pursued, at least collected, as new technologies are developed to help us better study all of these biological hallmarks, individual immune cells. We cannot simply study baseline measures. We have to address resilience for the reasons that I had mentioned. And there's remarkable multidimensional heterogeneity in everything with aging, but particularly involving the immune system, both at baseline in terms of responses to stimuli. But finally, please, 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 don't let the perfect become the enemy of the good. In other words, if gerotherapeutic trials try to do perfect biology of aging, they will never happen. If they ignore the biology of aging, they will not have any meaning. So we have to find some common ground between them. Thank you. I also want to give a shout out to, I neglected to include a slide to Graham Pavelets, who's been an important contributor to this and who couldn't be here today and has been working on this as well. Thank you.